These are some of the carbohydrates. Fine? We are surrounded by carbohydrates. Now the question is, can you tell me, where do we get the carbohydrates from? Do we produce it? Do we synthesize it? From where do we get the carbohydrates? Very easy. It's a result of the process of photosynthesis. The plants prepare it for us. The starch, it's again a carbohydrate. The glucose, it's a carbohydrate. Fine? So we get them from the plants, and the process is photosynthesis. Let's talk about the general structure of the carbohydrate. The general structure. At least 3 carbon atoms are there. At least multiple hydroxyl groups are there. Either they can have an aldehyde group, that is CHO group. Slowly try to understand. It's very important. Either they will have an aldehyde group, that is CHO group, this, or a ketone group, that is C double bond O group. Fine. Let's see some examples. See, this is glucose, which is an aldose. That means it has an aldehyde, CHO. And fructose, it's a ketose. It has a ketone group. See here, the ketone group. See here, the aldehyde group, CHO and C double bond O. Got it? This is the linear structure of glucose. You might have seen some circular structure of glucose like this, correct? We will talk about it. Today you're going to learn about it. Yes. So, glucose. This is the aldose. Fructose. It's a ketose. Got it? Can you see the multiple hydroxyl groups over here? Fine. Got it? Great. Between carbon, hydrogen, oxygen, it's 1:2, 2:1, isn't it? So the basic formula, can you derive the basic formula from it? Can I write CnH2n ON, or can I write CnH2O ON? Can I write this? So this is the basic formula, CnH2n ON. Fine. Just try to put 6 in case of N and try to find out if you're getting the formula for glucose. C6H2 multiplied by 6, that means it's 12, and oxygen is 6. So C6H12O6, this is the general formula for the carbohydrates. Fine. Carbohydrates, see, this is the general formula. Fine. Okay. The rebels. There are some exceptions to this general formula, and they are the rhamnose. You see the formula, C6H12O5. Come on, just count it. Is it correct or not? Count it. 1. These points are carbon. 2, 3, 4, 5, and this is 6. Correct? H12. 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. And O is 5. 1, 2, 3, 4, 5. Got it? Deoxyribose. The formula is C5H10O4. So these are some of the exceptions to the general formula for the carbohydrates. Clear? Remember, this question can be asked and question time. Great. Let's see. The most abundant biomolecule on Earth, carbohydrates, are synthesized by what? We just discussed about it. Come on, answer it. Let's see who can answer this question quickly. Let's see who can answer this question quickly. The most abundant biomolecule on Earth, carbohydrates, are synthesized by? The answer is C. Some bacteria, algae, green plant cells. Correct? Okay. Now, classification of the carbohydrates based on the number of the monomeric units. Number of monomeric units, based on that you can classify carbohydrates as monosaccharides, disaccharides, oligosaccharides, and the polysaccharides. Fine. Mono, di, oligo, and polysaccharides. Let's start our discussion with the monosaccharides first. Monosaccharides. Mono means single, saccharide, sugar. You know about it. Fine. Take it slowly. Understand it slowly. Mono, single, saccharide, sugar. Well, simplest carbohydrates because these are the monomeric units. It cannot be broken down into further smaller units of sugar. These are the smallest unit. Fine. They're generally sweet in taste. These are the simple carbohydrates. It cannot be further hydrolyzed into smaller sugars. Building blocks of large carbohydrates. Simple, very logical because these are monomeric units. I am so sweet. Yes. Can any one of you tell me what is the structure? Which carbohydrate the structure denotes? Well, let me draw a face around it so it will look better. Right? Okay. So, I am so sweet. What carbohydrate is this? Answer it. We'll figure it out as and when we are continuing our discussion in the session. Fine. The monosaccharides, based on the number of carbon atoms, monosaccharides can be triose, tetrose, pentose, hexose. They can be beyond that also. Fine. Triose, three. See it here. Have 3 carbon atoms. Glyceraldehyde is an example. Glyceraldehyde, okay? See carbon 1, 2, 3. Correct? Do you get it? Okay. So, 3 carbon? Okay. Now, the glyceraldehydes, 2 forms. Pay attention to this. This is very important. L-glyceraldehyde and D-glyceraldehyde. L-glyceraldehyde and D-glyceraldehyde. Here you have to understand something which I'm going to talk about, and to understand that you need to know the working of a mirror, plane mirror. You know about the lateral inversion that happens in a mirror? Look here. This is my finger. This is my palm, right? This is my hand. These are the fingers. Now let's imagine there is a mirror over here. This is the mirror. This is my hand. Try to figure out. Pay attention closely. Then only you'll understand. Now, if I try to look into the mirror, see it here. This thumb is towards my right. Correct? This thumb is towards my right. Here is the mirror. I put it like this. Where will it appear? The mirror will show something like this. Correct? Now, see, if I just try to figure it out, now here, the thumb is towards my left. These are the mirror images. Correct? Now, this one was 1, this one was 1. The same object gives this type of an image from a plane mirror. If I try to impose it, is it superimposing? No, because this thumb is here, this thumb is here. It's not superimposing, correct? These are non-superimposable mirror images. Are you clear about this? Again, once more. This thumb is this side. In the mirror, you will see it this side. This is the property of the mirror, lateral inversion. The right shows left, left shows right. Lateral inversion. Now, if I try to impose it, superimpose it, it won't get superimposed. So, these are non-superimposable mirror images. You'll understand why I'm talking about this. Yes. So, L-glyceraldehyde and D-glyceraldehyde. You know how this is named? You have to look into this carbon over here. This carbon, okay? See, L-glyceraldehyde is named when the OH group, slowly pay attention, the OH group is towards the left. OH group is towards the left. And here, D is when it is towards the right. So how will you remember? L for left, the other one is right. Fine? So, L-glyceraldehyde is when the OH group is on the left side, and D is when the OH group is on the right side. Fine? Try to imagine when you're looking into the paper. When you're looking into the paper, then how do you look at it? Fine? So D and L, glyceraldehyde. Now, I explained to you about these mirror images. Just take some time and try to relate. Do you think that these are the mirror images of each other? Yeah? Try to look into it. See, OH was this side here, like my finger, and here OH becomes this side, like my thumb here. Correct? So can I say these are the mirror images? And if I try to superimpose them, I'll just rub it, and then you can understand the superimposition. Try to superimpose once more. It does not get superimposed. Yes, so these are called enantiomers. Write it down. Enantiomers, okay? So non-superimposable mirror images. These are called the enantiomers. Got it? Well, so monosaccharide, you understand about the D and the L forms. Two optically active forms. Don't worry about this word optically active. I'm going to explain it. Just wait for some time. Two optically active forms, non-superimposable mirror images. See, here's a mirror, right? See the green one up, that's fine. But try to locate the other one. This one is here, this side, but the red appears on the other side over here. So these are non-superimposable mirror images. Now what we'll try to do is understand about these optically active forms. Let's see. Optically active forms. Now, light. If you talk about a source, let's imagine this is the source of a light. It's a torch. If I just light it, light is moving from this direction towards you. Fine. Light is moving from here to this side. This is the path of movement of light. Now, the molecules here vibrate in various different directions, but at a 90-degree right angle compared to the direction in which the light is traveling. If the light is traveling like this, then one angle can be like this. Okay, this side. Got it? Clear? Now, light in this way vibrates in different planes. Yes, that's a better word. They vibrate on different planes. Now, so I can just try to show you. For example, this is a source of light. So this direction, this direction, this direction, this direction, this direction. And the light is coming towards me right now from the screen. Okay, let's imagine from the screen, just look at it. It's going towards you from the screen. Fine. And it's vibrating in all these planes. Now, if here, let's now just turn the direction. Let's imagine the light is moving this side. Okay, many planes the vibration is going on, and I put a polarizer over here. Polarizer. You know what is the function of a polarizer? Polarizer basically cuts off all the directions, all the planes, and only allows light to vibrate in one plane. Fine. So that means if this is the slit of a polarizer, then only the vibrations of light which is happening along this particular slit will travel. So here it was here, this side, this side, this side, all sides over here. Correct? Now, once it passes through this polarizer, only one plane of vibration will be seen. So this is a polarized light and this is a non-polarized light. Got it? Do you want me to repeat once more? Okay, fine, no problem. The light vibrates in different planes. Now the polarizer cuts all planes and allows only one single plane to pass through. Fine, one single plane of vibration to pass through. Now if, for example, if this polarizer here, the slit is aligned in this direction, then only the vibrations which are going in this direction will be able to pass. So after passing from the polarizer, all the planes are cut off. Only one single plane of vibration of light is allowed to pass through. So this is now a polarized light. Here it was non-polarized light. Correct? Okay. Now, optically active molecules are those which can rotate. Yes. Which can rotate these polarized light. They have that capability. Fine. Now, there are two ways. Dextro-rotatory. Okay. Dextro-rotatory. That means, for example, now imagine. So here was a polarizer. Let's imagine I am the polarizer. Okay, the light is coming from behind me. So after passing through me, only this plane is there. Okay, imagine this is the vibration, 90 degree, correct? Only this plane is here. Now, dextrorotatory means if the molecule of a particular compound is here, fine, and it is optically active, then what will happen? It turns the plane polarized light, this is a polarized light, to the right or clockwise direction. To the right or clockwise direction. It's towards me. So if I talk from my perspective, it is towards the right, correct? This is my right. If it turns like this, if this light which has come, I'm the polarizer, right? It has come out. Now, if it turns right, that means these are dextrorotatory molecules. This is dextrorotation, right? If it turns towards the left, then it is leviorotation. Got it? That is anti-clockwise. Clock moves in this direction, correct? That is clockwise is dextro and anti-clockwise, that is towards the left, is the levio. Got it? So that is basically optically active forms. So what are the optically active molecules? The molecules which can turn the plane polarized light to a particular direction. If it's dextro, then it's towards the right or the clockwise. Sorry, right or the clockwise. And if it's left, it's anti-clockwise. Correct? Got it? Clear? Fine. These are non-superimposable mirror images. Relate to this. It's non-superimposable. Enantiomers. Great. Tetros. Let's see. Have 4 carbon atoms. They have 4 carbon atoms. Fine. D-erythros. Can you see? Okay. D means? You understand? So here, if you take this carbon, fine. D will be, OH will be towards the right. Yes. This OH is at the right. So, have only 4 carbon atoms. Can you see? 1, 2, 3, 4. Pentos, of course, they will have 5 carbon. 1, 2, 3, 4, 5. Next. Can you see? 1, 2, 3, 4, 5. Got it? Okay. So, if I talk about the ribose versus deoxyribose, DNA and RNA. Yes, in DNA there is deoxyribose, in RNA there is ribose. Fine. Let's see. Let's figure out the difference. See, the number of the carbon is labeled over here. This is carbon number 1 and this is carbon number 5. Carbon number 1, carbon number 5. See, ribose, in ribose, try to figure out the difference. Where is the difference? See the difference. It's in the second carbon atom. See, the second carbon here, they have OH group is there in ribose. But deoxy, oxygen is not there. So here, see, no OH group. Basically, I can say oxygen is not there. That's the reason it's deoxy. It's bound to H and H. It's bonded to H and H. Here it's bonded to H and OH. That's the difference between the ribose and the deoxyribose. Got the difference? And that you have to remember is at the second carbon. Second carbon. Fine? You know, this was a linear representation. This is a cyclic representation. Fine? See. Now again, try to relate. This is carbon number 1. This is carbon number 1. So see here. Okay? After the cyclic form, this is carbon number 1 and this is carbon number 5. Here it is 5. Fine? Got it? So can you figure out the difference? It's in the second carbon. Got it? Okay. So pentose. Now hexose is, of course, it is 6. 6 carbon atoms. Very common example, the glucose. Glucon D. Love it. See, this is glucon D, and now let me show you how the linear structure transforms into the cyclic structure. Okay, see here. There's an attack which happens between this OH and here. Okay, at the first carbon and from the 5th carbon. Similarly here also, L-glucose and D-glucose. Because of this, remember, 1st and the 5th, this structure comes up. This structure, see, the direction is different over here. Correct? 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. See the direction? Okay, great. Fine. See how it has transformed from the linear structure to the cyclic structure. Got it? Okay. Well, do you see? Are they superimposable or are they non-superimposable? Give me the answer. Are they superimposable or are they non-superimposable? Yes? Of course, they are non-superimposable, correct? Yes. See, this is a left-handed sugar. Why? See how it has formed. And this is D is the right-handed. Correct? See, D here, the OH was on the right, and here it is on the left. So D and L. Well, so glucose, fructose, these are some of the examples. Fructose you will get in the fruit sugar. Okay, so glucose and fructose. Try to figure out the difference. Here is a aldehyde. Here we had seen it already, the ketone. Ketone group, aldehyde group. Galactose. Can you figure out the difference between glucose and galactose? Quick! At which carbon atom the difference is there between glucose and galactose? Very similar structure. Label it 1, 2, 3, 4, 5, 6. Then only you can answer me. Which carbon atom? Come on, quickly. Quickly, which carbon atom? I can find the difference at the fourth carbon atom. See, here the OH is towards my right. Here the OH is towards my left. That's galactose. Fine? That's the only difference. Glucose and galactose. Well, now I'm going to talk about something very important, and that's the reducing and the non-reducing sugar. Fine? It's a bit of extra chemistry over here. Don't worry, I'll explain it to you. Well, so reducing sugar means those sugars in which there is free aldehyde and ketone group is present. Free aldehyde and ketone group is present. Don't worry, I know you cannot see here something free, but they are actually free. I'll show you. Non-reducing sugars are those which do not have free aldehyde and ketone group there. So no free aldehyde or ketone group. Fine? Okay. All monosaccharides are reducing sugars. Fine? So I'm sure you are finding it difficult to find the free, because the examples I'm showing here are all reducing sugars. But the question that you should ask, I'm sure you are asking, that I cannot see where is the free aldehyde. Note here. See here. Fine, carbon number 1. So these are basically the groups when the molecules are put in water in an aqueous condition. What happens? Try to note, I'm just showing a part of it. So this bond breaks. CH, OH, and this is bonded to C. Here it will be H. Here it will become OH. Fine? Got it? So because of this, is a free aldehyde group. You understand? So once more. So when this is put in water, what will happen? Okay. So when this is put in water, what will happen is OH and this bond which was there will break. Now here is a free aldehyde group. Correct? Fine? Yes. Similarly, you can try this with the fructose and the galactose also. You'll find at this carbon, it is free. But here, you won't find that free aldehyde group. But on the first carbon, you'll find the free aldehyde group. That's the reason they are reducing sugars, because they are able to reduce other molecules. Fine? We'll do an experiment to show that. Great. Are you clear about this? So what are the reducing sugars which have free aldehyde and ketone group? How will you understand? Here is the O. Here is also the O. Correct? carbon bonded to this oxygen, carbon bonded to this oxygen. These are the groups which when put in water, okay, this bond, and here this bond, it breaks. It results in the formation of a free aldehyde that reacts. Correct? Okay. So all monosaccharides are reducing sugars. Remember this. Reducing or non-reducing? Do you want to do a test? Okay, let's do a test. I have the reagents to do the test. Fine, let's perform the test. And I'll just give you a brief about the test. So we will use Benedict's solution, and what you'll see is something like this. Fine, let me do the test for you. Well, before that, I'll try to take some quick protection so that I don't hurt myself, as I told you at the starting only, or rather I don't stain myself with the colors of the reagents. Great. So you understand, right, what we are going to do now? We are going to detect what? The reducing and the non-reducing sugars. Fine. So we will require a reagent, that is the Benedict's solution. Fine. Yes, and I have the Benedict solution over here. Correct? Well, so let me see. Here I have the test tube. Now what I'll use is some glucose. Okay, I'll use some glucose over here. Let me pour some glucose. I'm using simple glucose. Okay, you can also do this test, but of course, when someone experienced is present in front of you. So see, I've taken some glucose, right? Okay, great. Come on, you go away. Your work is done. I'll prepare a solution. Glucose solution is ready. Do you see the color? It's like simple. I am sure you know about this color of this solution. Correct? Well, I will use one more where I am going to use some sucrose. This is table sugar. It's sucrose, correct? Let me put some sucrose over here. Yes, I have sucrose and I'll prepare this sugar solution. Okay. I'll just shake it so that it gets quickly dissolved. Come on. Okay, now what we'll do is I have these two and I need this guard so that things are properly and clearly visible for you. Fine? Okay, can you see? Here, this is glucose. You know what? I should write it down so that I don't forget what are the solutions. This is glucose. This is sucrose. We'll study about sucrose in a while. So wait. Now, see here. This is glucose, this is sucrose. Now what I'll add is the Benedict's reagent. You know, this reagent has copper sulfate. Can you see the color? But I'll try to do it. Don't fall. It's blue. Copper sulfate, it's blue. Now, see, I'm adding it. Okay. So here, you know what? I told you about the reducing sugars. Copper sulfate, this Benedict solution is basically a source for the copper. Now the copper, if the sugar is reducing, then the copper, what will happen? The copper will get reduced. Copper sulfate serves as a source for the copper ions, this Benedict reagent. Fine, you see, it's blue. Okay, let's just shake it a bit so that we get the solution properly. And it has to be heated a bit. Okay, it has to be heated a bit. And can any one of you tell me what is reduction? What do you mean by oxidation? What do you mean by reduction? Because that is important. In the meanwhile, I'll just shake it so that it quickly gets dissolved. See the color? Blue. Correct? Yes. You know, the process by which the substance loses an electron, if a substance loses an electron in a chemical reaction, it's called oxidation. If it gains electron,